Hello, everyone. My name is Renee Napoliello. I'm a PhD candidate at the Quan Lab at the University of California, Davis, and today I'll be presenting on my project utilizing convolutional neural networks to predict CRISPRi efficacy. So to begin with, what is CRISPRi or CRISPR interference? Well, as the name suggests, it's a tool used by molecular biologists to repress expression of a transcript or gene of interest. It works similar in some of the subcomponents to the more well-known CRISPR-Cas9 system. You have a guide RNA that acts to target your complex to your region of interest on the genome, in this case, the promoter or gene body of the transcript you would like to repress. A Cas9, in this case, a deactivated Cas9, so no double-stranded breaks will be induced. And it works as sort of a pack mule to bring in the tethered transcriptional inhibiting protein that works to repress expression of the gene. So the challenges in the field is something I'm quite familiar with because my previous PhD project was actually involved a CRISPR-based epigenetic editing lab. And I was tasked with using CRISPR-A, which is the opposite of CRISPR-I, to activate expression of a gene of interest to treat a rare neurological disorder. So I'm about to sum up 3 years of work in one slide. But don't worry, you're not going to be here that long. So I took 3 different libraries and screened 25 different guide RNAs in a very cheap and easy to use cell line. I was mostly satisfied with the results. I found several guides that activated expression. And then when I proceeded to neural stem cells, which was a better cell model for my disorder, I was disappointed to find that some of the guides I had found in my screen no longer worked. And even the ones that did work weren't activating to the same degree that they were in the previous cell type. It took me another few months from there to validate a single guide RNA in multiple patient-derived cell lines. So including all the other factors that go into running wet lab experiments, this ended up being a 3-year project with a lot of hurdles and frustrations, but it really got me interested in the field because I wanted to understand, why am I seeing these differences? Why is CRISPR activation working in one cell type but not another? Intuitively, it makes sense. Transcriptional regulation itself is a very context-dependent mechanism. So of course, epigenetic-based perturbations might also be related to the context of the cell type. So not only is it an interesting question to probe from the basic science standpoint, but I can also test to the fact that understanding how to improve efficacy of these interventions can also significantly reduce time and resource costs at the bench. So the general overview of this project is to combine the hypothesized input modalities that we believe contribute to understanding CRISPR-I efficacy. So the first modality is epigenetic signals and feature annotation. So these are things like chromatin accessibility, ChIP-seq signals for transcription factor binding, location of the guide and the transcript, etc. The second modality we wanted to use was based on the genomic sequence, in this case, the region around the loci we are targeting. And the third modality relates to the RNA itself. The RNA, not only the sequence features, but also treating it as a functional unit of this RNP, which is related to expression, thermodynamics, secondary structure, etc. So as a broad overview, all 3 individual inputs receive their own independent convolutional blocks before meeting in the middle of the model for a multimodal integration, and finally landing on a singular output that is some measure of guide efficacy. So in terms of the data sets, we wanted to start with 2 different inputs and 2 different outputs to test the robustness of our model architecture. So the first is a dual guide screen. So the dual guide screen pairs guide RNAs so that every promoter will have 2 separate CRISPR-I complexes targeted to the same promoter. However, across promoters, they only have one option, i.e. there was only one guide pair per promoter. The single guide screen was a bit different. You only had one CRISPR-I complex per promoter, but you had 10 to 20 options. Essentially, this experiment was tiling guide RNAs around a promoter to find the optimal site. They also differ in output. The dual guide screen had a direct readout where it analyzed with Perturb-seq, which is a modification of single cell RNA sequencing, the fold knockdown of the transcript of interest. The single guide screen was an indirect readout, so they looked at relative cell death and growth as a measurement of the degree of repression of the survival gene of interest. So the first question we wanted to tackle was the most basic idea around CRISPRi, which is where is the guide in regards to the transcript? So we tested out 3 different ways to represent this input. The first way was the most obvious, which is explicit annotation of the transcript track. The second way to represent transcript was fixing the orientation of the windows. When I say window, I mean the input for the convolutional block. So that regardless of if the transcript is positive or negative stranded, left-handedness and right-handedness would always equate to promoter direction to gene body direction. And finally, we wanted to see if centering the context of these windows, or literally centering the midpoint on the TSS rather than on the guide itself, would better help the model understand transcript location with regards to guide. So for the single guide screen, you can see a very apparent trend immediately, and that is the inclusion of transcript annotation, which is grayed out that bottom row in the table. A significantly improved model performance. The readout we are using to assess model performance is a Pearson correlation between true and predicted output for 5-fold cross-validation. You can also see for the single guide screen that centering windows, input windows, on the guide itself instead of the transcription start site actually improved model performance as well. You see some similar trends in the dual guide screen. So again, explicit inclusion of the transcript annotation significantly boosted model performance. Something interesting though, in the case of the dual guide, orienting the windows in regards to the strandedness of the transcript also essentially provided a helping hand to the model, when in the single guide screen that really had negligible impact. So now that we had a model learning from transcript location and guide location, we wanted to understand what the model was learning, and the biological relevance behind that. So to interrogate that, we generated a synthetic input where we tiled guide RNA positions 1 base pair apart for the entirety of the window, meaning 1,000 base pairs upstream and downstream of TSS, and mapped out the predicted output based on the model. So you can see here that the model exhibits preference for guides landing within the gene body, as well as a significant preference for immediately downstream of the TSS, and a depletion in activity upstream of the TSS, as well as some periodicity, which is really exciting to see because it seems to line up with a lot of some empirical studies about not only CRISPRi efficacy, but nucleosome phasing. I can't go into that right now. We also just wanted to assure that these predictions were based on ground truth values. So we plotted out the distribution of the training samples to see that they were tiled around the entire input window. The dual guide screen, representing that was a bit more complicated because instead of landing a single guide along a transcript and testing output, we had to consider pairs of guides and all possible combinations. So to represent that, on the right-hand side, we have a heat map showing the predicted fold knockdown depending on guide A versus location versus guide B location, the lettering not actually mattering. And you can see, even though we have a different experiment and a different type of output, there are similar trends to the single guide screen, namely that the model exhibits a bias towards guide RNAs, both the members of the pair landing within the gene body, a depletion activity immediately upstream of the TSS, a spike in activity downstream of the TSS. The graph on the lower right-hand side is a depiction of the marginal distributions, so for all guides within a pair. So just another way to look at this heat map, similar to the single guide screen. And you can see some similar trends to the previous screen I showed. So now that we had established a way to show transcript annotation and guide annotation, we wanted to focus on the RNA input. So before I proceed, something to mention about guide RNA is that there's 2 subcomponents. There's the scaffolding region, that is the point of interaction between the guide and the Cas9, and there's the protospacer region, and this is the targeting unit. This is what's complementary to your genomic region of interest. So the protospacer will change between guides, the scaffold sequence will not. So we wanted to see if including the scaffold sequence at all was important for a model to make predictions based on guide efficacy. And you can see here that the scaffold plus protospacer, so that's the blue box, that input actually did improve the correlation between true and predicted output. So just a reminder, even though scaffold sequence is identical between samples, metrics related to secondary structure, opening energy, that can all be different. So now we wanted to understand, okay, if the model is learning from RNA now, what is it? What are the rule sets it's taking away from that? So we ran the guide RNAs through the model trained on RNA features and generated the top 10% and generated a consensus structure to see, what does a top performing guide look like? And then we shaded in the nucleotides based on the the highest absolute value of integrated gradients from feature attribution. So that's a very long sentence to say, we generated a consensus structure of the guide RNA, and then we shaded in the nucleotides the model was paying most attention to when it came to guide RNA folding. And you can see it is paying attention to some of the nucleotides related to stem loop formation. Since a part of our feature input was protospacer sequence, we then took bottom and top performing predicted guide RNAs and wanted to see how the model was making decisions based on sequence motif. And it's interesting to see that the significant preference in guanine enrichment and uracil de-enrichment in the top performing guides, is that rule is a lot softer for the bottom performing guides, even though uracil is not the most enriched nucleotide. It is much more present in a low performing guide than a top performing guide. So now that we had an annotation-based input head and an RNA feature input head, we wanted to see if combining these modalities would actually improve model performance as suspected. So this graph is showing the single and dual guide screen, and you can see that the single annotation input versus the RNA individual RNA input for both screens performed worse than combining annotation-based and RNA-based model. We also tested out DNA input at this time across different models and parameters. It had negligible impact. So moving forward, we did not focus on that input head. So now that we had RNA-based, annotation-based input, the next step, which is really exciting, is focusing on epigenetic signal tracks. So for the dual guide data set, we tested all possible pairs of 8 candidate signals that are related to chromatin accessibility, promoter activity, et cetera. And we then made a heat map showing the largest impact or most significant impact on correlation between true and predicted output. And the standout signals were DNA-seq, RNA polymerase, and to a lesser extent, HDAC1 as well. And as a way to interrogate, based on epigenetic signals, what the model was learning, we applied the similar logic of grouping bottom performing and top performing guide RNAs and took essentially snapshots of the windows they were landing in to compare the relative locations of epigenetic signals. We noticed an interesting trend where top performing guide RNAs landed squarely within DNA-seq peaks, so chromatin accessible regions, whereas they seem to land outside of those peaks for low performing guide RNAs, which is consistent with previous literature on chromatin accessibility and CRISPR-based interventions. So in conclusion, our convolutional based approach is able to combine multiple input modalities relevant to CRISPR-I intervention. In addition, our hypothesized input of epigenetic and feature signals, as well as RNA features, seems to provide essentially a way to predict CRISPR-I efficacy in regards to guide RNA. Finally, our unbiased assessment of RNA features, especially secondary structure, suggests that RNA folding may play a larger impact on CRISPR-I intervention than previously believed in the literature. And of course, I'd like to finish with an acknowledgement for everyone in the Quant Bio Lab for helping facilitate my transition from the wet lab to the dry lab. It's been quite a journey. I've enjoyed every day, and I'd also like to give a special shout out to all the pictures of my lab mates up here who are co-authors on my abstract, as well as Dr. Gerald Kwan. Thank you.
We have time for one question.
I feel like I'm asking a lot of questions, but great work. What do you think the implications of this are for already done CRISPR screens with all kinds of different guides, or even like smaller CRISPR experiments where you compare a whole bunch of guides here versus a whole bunch of guides that may have all these different secondary structures and sequence compositions, right? In terms of comparing within one experiment, the effect sizes, I guess we all ignore any of these things that you're talking about right now.
So I believe you're asking, what is the relevance of studying previously done screens? Is
no,
that
what all these results that you're finding suggest that there is a lot going on the guide RNA design.
Yeah.
Right. And then we all use like multiple papers and talks here. We use these effect sizes from these CRISPRi screens to train our models, to test our predictions, and so on and so forth. So what does this imply going back? Going forward, I think the implications are clear. Going backwards to the existing data.
I see. So I think the implications are, what's been well known at the bench is that the CRISPR-based interventions are very, difficult to predict. So understanding all the factors that weren't previously understood about what makes a guide ineffective allows us to look at screens performed in cell types different from what the libraries were originally designed on, and we're able to understand, the reason we don't get a signal for perturbations at some promoters may be because they are simply ineffective. Our tools were ineffective rather than the tools worked, but changing gene expression didn't do anything. So I think that's one thing. And I think also going back to all of these screens, especially, diverging from survival genes and going to other promoter types and really delving into, how epigenetic signals relate to genetic perturbations. I think that can also help us understand and classify, the essentially different promoters and how they behave differently within a cell type, but also across cell types. So those are two things that come to the top of my head that I'm excited about.